And also, if you'd like to participate within the BioVis community and play a more hands-on role, please come and chat with me after the session. And today I'm going to talk to you about a tool that we have developed in my research group at Icahn School of Medicine at Mount Sinai in New York City. It's called ClusterChirp, a GPU-accelerated web platform for AI-supported interactive exploration of high-dimensional omics datasets. And before I begin with ClusterChirp, I want to give you a little bit of information about what we do in my research group. We are a computational cancer genomics lab that aims to identify and target drivers of cancer genesis, progression, and therapy. And within that, here's a word cloud of some of the words that we most frequently use in our abstracts. And you can see tumor, cancer, network, etc. And for this purpose, we develop software, web interfaces, and ClusterChirp is just one of the tools within this environment. And we collaborate with clinical, experimental, and computational teams. And some of the consortiums we participate are HIPC, CPTech, and CMAC-CIDC that are supported by the NIH. And within our team, we want to be able to build bridges between DataVis and cancer, clinical oncology research, basic research, computational oncology. We interact with these teams, and we also do web development and visualization design. ClusterChirp, the motivation for developing this tool, ClusterChirp, is that in computational biology, often we visually inspect tabular omics data to find patterns, trends, and anomalies, which may be key to new biomarkers or therapy targets. And as the omics data sets grow in size and modality, traditional tools struggle with their visual displays. Some of the issues are computational speed and memory limitations. Large matrices can be an issue if there are more than 1 million or more cells within the data set or data points. Some of the user interfaces are complex, and they have steep learning curves for non-computational users. They have slow processing time when the data gets large, and clustering and visualization on the fly is not always possible once the data set kind of blows up and is very large. And also there is somehow limited room in manual and biological interpretation has to be done manually. There's no connection to other tools or biological databases with a lot of these tools. And within ClusterChirp, we wanted to address some of these issues, and we decided to develop a GPU-accelerated web platform which enabled on-the-fly clustering, on-the-fly interaction with massive data sets. And we haven't hit our upper limit yet, but we've tested it around 10 million data points or more, and it can be quite quick, and it gives us the on-the-fly results. And it does have an AI-powered natural language interface, so if you don't want to interact with the tool through the keyboard, you can just write to its chat box, and that's the name Chirp, because you can chirp along with it. And it has a real-time interaction and parallel computing capability. It's fast, responsive heat maps and correlation networks visualizations. And we've also integrated a preliminary pathway analysis just to do on-the-fly quick analyses and enrichment. So this is the flow chart of ClusterChirp, and so the user interacts with the tool either through the keyboard or the AI chatbot, and once they upload their data, they can do a lot of the usual interactions: zoom, pan, sort, cluster, etc. And in the backend visualizations, we use DeckGL, D3.js, and React Force Graph. And some of the functions that we can also use is hierarchical clustering, normalization, and also using ChatGPT. And in the back end of that is Python, Django, Scikit, NumPy, Pandas. So the output is either a heat map that user can completely interact with, or also you can, after you cluster it, depending on your priorities, you can also look at the clusters, individual clusters, more detail in network views. So this is a snapshot of the ClusterChirp homepage, and here we have the main heat map, and then you also have the overall, because these matrices can be large, the little box here shows you the overview, and the user uploads their omics data to visualize instantly, and the AI chat box is in the, I don't have a the AI chat box on the bottom provides like the capability to chat with it, and I'll just go ahead with a demo. So once you so this is the landing page, and then here's the how the input file format is, and you can just like upload your data, including your metadata, into cluster into our tool, and you can zoom in, look at the individual cells, zoom out, and look, because it's a massive data set, you can look at different areas of the heat map depending on which area is potentially of interest to you. You can choose different metadata to look at. You can order it with respect to sex, for example, here at the top, or histology, or you could sort it with respect to sum or variance, or you could just go ahead and cluster it, and all the operations are interactive and they're done on the fly. And here you see different clusterings. You can cluster the rows, you can cluster the columns in different ways, and then you can look at the metadata to see details. For example, what are the cluster memberships, individual cluster memberships of the clusters. You can see through those. And you can also search for a specific gene that you're interested in, and it zooms directly to that gene, and it locates where exactly it is in the heat map. And you can make it darker, you can make it lighter, depending on what you want to see, if it's going to help you with your analyses. And you can change the metadata that you're interested in. For example, now we have like 3 metadata types on the top, but you can add or subtract any metadata that you want. And again, it's updated on the fly. And let me see. Yeah, so that's pretty much it from the landing page. And then, oops, sorry. Okay. And but you can do all of this actually without having to use your keyboard and mouse. You can just interact with the AI interface, and the command system is pretty flexible. You can type what you want it to do. It's not going to respond if you say hi, but if you tell it to interact with the heat map, then it's going to do what you want to do. So some of the tools you can say is filter top 50 high variance rows, and then it's going to show you that. Show only males within your cohort, it's going to show you that. Or it'll have custom criteria applied, for example, cluster rows and sort by sum. And so basically it can do complex operations. And you can also give it user-defined parameters. You can filter any number of rows and columns you need depending on what you want to do. So here's a demo that we can have to better understand. So that's the chat box, and we gave some prompts so that the user is not confused and can choose some of these prompts just to make the analysis easier. But this is not a specific vocabulary. You can use anything that you want, any operation that you want, and you just type it in there. And so So for example, you can do filtering, you can, so for example, select top 30 most variable genes if you have a transcriptomic database and you want to look at only the top 30, the most variable, and then you have it. And if you have like, let's say a cohort of 1,000 people and you just want the 30 genes in these 1,000 people, you have it on the fly right away. And you can also, for example, make it dark, make it light. You can also do that here as well. Any of the keyboard commands you can do right here. And again, it does everything on the fly. And and you can also, for example, only select the males in your cohort, for example, or people with a certain property. You can do that also through interaction with the AI interface. And yeah, so they're different. Okay. The another thing that we included is that you can also, if you're doing if you want to have a biological on-the-fly understanding of the biology, you can see if the genes or proteins in your experiment are enriched in a certain cancer pathway, for example. And you can check the pathway. You can say which DNA damage response genes, for example, are enriched in the study, and then it gives you right away only the genes in the DNA damage response pathway. You can say DNA damage response pathway, top 10 genes, it will give you that also. So you can also do pathway enrichment analysis on the fly. And another functionality that we added is that you can also, once you pick a cluster within the heat map, you can, and if you want to see how the nodes within that cluster interact with each other, you can look at them as a gene correlation network, and you can do that in 2D or 3D depending on what your needs are. And also you can do the pathway enrichment. We have include most hallmark pathways, and you can choose some of the pathways that are of interest. For example, Go, KEGG, Reactome, the most commonly used pathways. And so all you have to do here is to after you cluster or not cluster, ask to do a pathway analysis. And it also links out to other tools like Enricher that was not developed in my team, but it has a very detailed pathway analysis infrastructure. Or if you're interested, you can, or if you're interested, you can look at a specific cluster as a network, and the connectivity of the network of the nodes within that cluster. So you can look at the subclusters within that cluster, and you can do that in 2D. You can do that in 3D, depending on what is of interest to you, to get a better idea. And here we have both of these visualizations already implemented. And again, you can do the pathway enrichment analysis and specific pathways and which pathways are most enriched. You can have the p-values and see them right away on the fly, so that it will help you go to the next stage, but you don't have to go through multiple different tools in order to have an on-the-fly initial understanding of the data that you have. And yeah. So as summary, ClusterChirp helps biomedical researchers to extract meaningful insights from complex multidimensional omics datasets to accelerate discovery and hypothesis generation. It's a powerful web-based platform to visualize and interactively explore high-dimensional omics data through GPU-accelerated heat maps and clustering analysis for high performance, speed, and scalability. It's designed for large-scale genomics, transcriptomics, and proteomics data sets. But of course, if you want to use it for other cases, go ahead. It's just that you won't be able to do gene set enrichment, but everything else you can do. It enables users to upload, cluster, filter, and analyze data in real time. It has multimodal capability, which means it seamlessly switches between heat maps and correlation network views. And it's freely available at the ClusterChirp mssm.edu address. That's where it lives right now. Feel free to go ahead and try it with your tool. And if you have a data set that you'd like to try it on, and if you have questions, just let us know. It includes both the data set that you have, in addition metadata, so you can provide time points, time series, gender, ancestry, etc., any other metadata associated with the data that you have, and to be able to have meaningful discoveries. So its AI-powered language interface transforms the user experience, allowing researchers to generate complex publication-ready visualizations. Any of the visualizations we have, you can download them in different formats and use them in your papers. And yeah, and most of this work was done through a Cancer Moonshot grant from the NIH that I received, and it was developed by Osho Rawal, who's a web developer and senior scientist in my research group. And but on the other hand, a lot of the other team members have also helped, especially Sasha Ginyatic and Edgar Gonzalez-Kozlova from Mount Sinai. And with that, I'm very happy to answer any questions you may have. Thank you.
Okay, two very quick, well, we have a little time of questions. We're already running a little late. So you were first, I think. Robert?
Yeah. Very, very nice interface, and I think there are a lot of powerful interactions there.
Oh, thank you.
Zeynep, I was wondering, what's under the hood when you say like GPU accelerated clusterings? Like what kind of clustering algorithms are there? Can you choose different ones, or is there like a plug-in capability if I would like want to use the tool with my own clustering? Maybe you can elaborate a little bit on that.
Yeah, excellent question. So right now we just use hierarchical clustering because that's what a lot of people use. It's a very good point to be able to use your own clustering algorithms. I think we can accommodate that. So and the tool lives in our GPU accelerated server, so that's why we have the speed that we have. And yeah, it's possible. It's doable. And maybe we'll do that.
Thanks.
But promise to use it.
I promise.
Okay. Yeah, Pinky.
A very, very quick question here.
Yeah, I wanted to ask about the pathway enrichment analysis. So is the corresponding background set?
I'm just going to say to us.
Okay.
Yeah, I wanted to ask you about the pathway enrichment analysis. So is the background set of genes for the enrichment defined by all the genes in the annotation or by the expressed genes in the data set? Because this could make a difference.
Yeah, so that's an excellent point. Right now, it's just the genes that are within that cluster. And for the background, we use the full transcriptome. And this is not for an and for all. I don't know the exact term there, but it's just for on-the-fly, quick understanding of the data so that you can have an initial understanding of it. And but there are other tools that it links out to. If you want more detailed analysis, we linked it to this cool tool called Enricher that was developed by Avi Mayan's lab, and it's so detailed and it's so it's in one of the demos actually. So it's so detailed that there was no reason for us to reinvent the wheel, but we have in one of the demos. So it automatically takes the genes that you have, and all you have to do is to click on the button that says go to Enricher, and you can do the rest of the analysis in Enricher. And if you need to update the background, you can update it there. So yeah.
Yeah, thank you.
Oh, you're welcome.
Okay, so thank you very much, Zeynep.